Thank you, Samson. Thank you for the introduction, and thank you, Ellie, for the invitation as well. It's an honor to be presenting in Friday for Birds and to tell you about the work I've been doing, some of it at least, over the last 20 years or so on Thinkerbirds in Africa. Okay, so as Samson mentioned, I'm an associate professor at the University of Cyprus, and I have the work I'll be presenting involves a large research team, various members of the lab, past and present, are pictured here and named here, including postdocs, graduate students, and undergrads. Okay, so what are our research interests? We're interested in understanding what explains variation in visual appearance and also the acoustic communication signals of animals. So communication involves the sending of a signal from a sender to a receiver with the aim of obtaining some sort of response from that receiver. One of the questions that I'm often asked is, why Tinkerbirds? Well, there are a number of reasons why I started working on Tinkerbirds and continue to work on them. In particular, these two species that I focused most of my research attention on in the last 15 years or so, the red-fronted tinkerbird and the yellow-fronted tinkerbird. One reason is in terms of song, variation is unambiguous. Songs are simple in structure and they differ primarily in two characters, in speed and in pitch. Feather color is also something that's very easy to measure and to differentiate between species, as you can see here in red-fronted and yellow-fronted tinkerbird. And in these two species in particular, they interbreed where they come together, and that offers us many opportunities for studies, and I'll be talking about that in the course of this seminar. So characters of birds such as morphology, feather color, even song are dependent on both environmental effects and genetic effects. So I'm going to talk about those in turn. Looking first at environmental effects. Birdsong is an essential trait to accomplish important functions for animal survival, and its transfer across space is essential to ensure the prompt response of the intended receiver. Now, there are two. Seems to be stuck here. There are two major environmental factors that may affect the transmission of song. One is the transmission properties of the environment. As you can see pictured here, songs may be affected by the density of the environment. There's a lot of work on the acoustic adaptation hypothesis, and with the suggestion that songs are shaped to be more tonal in dense environments and dense habitats, more broadband in more open habitats. Also, noise is thought to be a major factor that affects the transmission of acoustic signals. Sound waves of different frequencies and bandwidths differ in how well they transmit through the habitat, but also loud and continuous background noise imposes selective pressure on birdsong to increase its effectiveness in noisy environments. The noise may mask that signal. So how do birds deal with signal interference? This is an example from some work that I did some years ago on green hylia, looking at how a bird is singing at a certain frequency in order to get its signal across. So here, in this case, it's a male trying to attract a mate and get its signal across at a certain frequency. And if there's no noise, it manages to get its song across and gets the desired effect. Now, working on green hylia song, we looked at how noise at different frequencies may affect the transmission of this green hylia song. So Murchison Falls National Park in Uganda, there was this continuous band of noise at around 7 kilohertz, which is the result of singing insects, in particular cicadas. Green hylia sings a song at around 3 kilohertz. It's not affected by this noise at the higher frequency, and so it can transmit its song to its intended receiver. All right, so we had a look at this, and we saw how our green hylia are singing at a lower frequency than the cicadas are singing at Murchison Falls National Park. So all's good there. But if we get that individual and they sing in another national park in Uganda, in Bwindi and Panchop National Park, well, there the cicadas are singing at around 3 kilohertz. And so if the green hylia sings at that same frequency, its signal will be masked. It won't get its signal across the intended receiver, won't get the desired effect in the receiver. So what appears to happen there? So interference with signal transmission can be overcome by changing frequency of a song to use a non-monopolized bandwidth. And so the bird will need to sing at a lower frequency or higher frequency than the insects are singing. What we found with green hylia was that there were these shifts in frequency in accordance with the background noise. Of course, green hylia is a bird that is a vocal learner. We'll go on to that shortly. So there is a lot of support for the effects of the environment on song. Birds of closed forests sing at lower frequencies. They sing tonal songs. Birds of open habitats sing more broadband repetitive songs. But studies within species may be less conclusive. There is much support for the effects of noise on song frequency. This is dependent on vocal learning with auditory feedback. Is that what might be going on with green hylia as well? That it can hear its song and hear the noise, and might have there might be plasticity in its song that allows it to adapt and learn to sing at certain frequencies. With regards to such studies on noise, there's a bias towards the study of anthropogenic ambient noise versus studies on natural ambient. So most studies focus on songbirds that have this vocal learning, this high vocal flexibility. There's much less focus on birds whose songs are innate. And so there's a picture of a yellow-throated tinkerbird, which we will argue is innate. And so another thing that we're often asked is, well, how do you know that tinkerbird songs are innate? Well, song learning has only been demonstrated in 3 orders of birds. That's in the Ossian passerines, in parrots, and in hummingbirds. Tinkerbirds are Pisciforms. So they're not in one of those groups where learning has been demonstrated, but that doesn't mean that necessarily that they can't learn this. We have some evidence, which we published on. Now, in birds that learn their songs, that learning process takes several months, sometimes years, before they can sing that adult song. Well, in these videos, we've got some videos of a nest of a yellow-rumped tinkerbird, and what you can see here is a nestling. Singing there from that nest hole. So I think that's pretty good evidence that the song is innate. This nestling's probably about 12 days old. So we've looked at the effects of natural noise here, abiotic noise on tinkerbird song. Here looking at noise from the ocean. In this study performed with Matteo Sebastianelli and Dan Blum's team, UCLA. And we investigated whether 3 species of tinkerbird adapt the frequency of their songs to increased ambient noise from oceans. So these are 3 species: yellow-throated tinkerbird, yellow-rumped tinkerbird, and red-fronted tinkerbird. And the study was performed, it was based on recordings obtained in Cameroon and also in Kenya and Tanzania, so West Africa, Western coast and Eastern coast, and different species. And so we got a number of recordings of these 3 tinkerbirds, and we also recorded ambient noise in Cameroon. And we hypothesized that the ambient noise would decrease the further away we got from the sea, that yellow-throated tinkerbird frequency would increase as the ambient noise levels would increase. We could only test that on yellow-throated because we only had those ambient noise recordings from Cameroon. And the frequency of the song would decrease the further away we got from the coast. And what we found was that song frequency did indeed increase with ambient noise. So we can see in plot A here, as ambient noise increases, so as these values of in dB approach 0, we see that the song frequency increases. Ambient noise is obviously a lot higher close to the coast, and you can see it actually drops quite dramatically as we go from 0 to 1 and then 2 kilometers. And here is the frequencies of the songs of these 3 species as we approach the coast. So as we go towards 0, we can see that there's an increase in frequency, which was significant in 2 of the species, in yellow-throated tinkerbird and in red-fronted tinkerbird, but it wasn't significant in yellow-rumped tinkerbird, which conversely has the lowest frequencies on the 3 species. Here's an example of a spectrogram and also a power spectrum of the noise that you can see from the ocean. So you can see an example here in A with how much noise there is at these lower frequencies, and that selective pressure you can see on the spectrum as well for, in this case, the yellow-throated tinkerbird to sing at a higher frequency so that it can get its signal across to its intended receivers. And here on the right, we see the 2 songs of the yellow-rumped tinkerbird and red-fronted tinkerbird and how they're affected by the background noise there. Now, what you'll see here is, in this case, yellow-rumped tinkerbird, this is the coastal form fisher-eye from coastal Kenya, which actually has this trill-like song. And one of the ways that birds may escape noise is by having a much more repetitive song. It could be that that's better adapted to those conditions. Also, there might be competition for frequency between the 2 species. So if it's any higher in frequency, it will start overlapping with the frequency of the red-fronted tinkerbird song. And that's another thing that we've looked at quite a bit as well, is how different species interact and how that might affect the frequencies of their songs. In fact, we've done a lot of work on interactions in tinkerbirds, and we've focused on in the forest zones of Africa on yellow-rumped and yellow-throated tinkerbird, and in the savannas on red-fronted, yellow-fronted tinkerbird. And so this is going back to my PhD work on tinkerbirds and looking at the songs and how these 2 species of yellow-throated and yellow-rumped tinkerbird interact. So this is what a yellow-throated tinkerbird song sounds like. Yes? Oh, it works. Okay, great. Yellow-throated tinkerbird. And so you heard how similar that was to what I just played before for the nestling. Very similar. So again, here you have 2 songs that have a similar structure, but one is quite clearly faster than the other, and there may be a difference in frequency as well, as there is in the spectrogram that I show you here. Well, we examined, all right, now it's going to play a lot. There, you can hear them both together. Okay, so we examined songs of these 2 species in populations where they occur together, and also populations where you find only one species or only the other species. So that's on this map here, and this is in Cameroon, Uganda. And what we found was that where they occur together in sympatry, the songs are more different in frequency than where they occur in allometry. And this was something that we, with some experiments, showed facilitated species recognition. They could tell the differences between the two species with that greater difference in songs. The patterns we found here were also connected to differences in morphology, and morphology varied along an elevation gradient. And so, yeah, so we controlled for the effects of the environmental gradients in these analyses. But then the question is, can we predict song frequencies at continental scales across environmental gradients? There are certain predictions that we might be able to make because there's this well-known relationship between body size and latitude that's linked to ambient temperature, which represents a major constraint in endothermic animals. And this is known as Bergmann's rule. As endotherms in particular approach the poles, these higher latitudes, body sizes of a lot of these endotherms are bigger, so they have a larger body volume to body surface area ratio. And this relates to retaining and or dissipating heat. Another rule, ecogeographical rule that related to morphology is Allen's rule. With this one, Allen's rule posits that appendages such as tarsi in birds, tails, and also the beak will be larger relative to body size to help with heat dissipation towards the tropics. And both of these might have an effect on song frequencies. Does temperature-dependent variation in morphology affect variation of other traits at a macroecological scale? Well, there is a well-known relationship between song frequency and body mass. This is in pasturing birds. So this is across some 5,000 species in a study done by Mikula and colleagues. And it shows that as body size increases, song frequencies are lower. This is something that's also been shown within species, actually, in a study that we did in Mexico on Mayan anthrash. We found the same pattern in both males and females. Beak length should also be correlated with lower song frequencies, since the vocal tract, which includes the beak cavity, has resonating properties that can affect song frequency. And so this leads us to certain predictions. If we look at a mean annual temperature map here of Africa, we would predict, as we did here on the right, that as temperatures decrease, body sizes will increase at these higher latitudes and elevation, and as a consequence, song frequencies should decrease as we approach the poles or go up into the mountains. With Allen's rule, the prediction would be that as temperatures decrease, the beak length, the relative beak length would decrease, and then as a result, the song frequencies would increase. And so they're opposing hypotheses in relation to song frequency. In order to look at this question, we used a large data set collected over a number of years, including field-based measurements from fieldwork that we performed in B, measurements that I obtained from museum specimens in A, and also a large number of recordings, mostly our own, but also some from online repositories across Africa. And that was in these 4 species of Tinkerbird. And so we then tested for Bergmann's rule. We looked at the effects of latitude, elevation, taxon, because there could be differences among the species, and also vegetation structure and density on body size. Similar predictions on the relative beak length, but also because it's relative, we control for body size, and also on song frequency. And here's a distribution of raw data in B and C here. In B, it's the morphology. So this is after principal component analysis. PC1 explained 63.5% approximately of variation in body size. And what you can see here is larger individuals are at these higher latitudes. And we see that body size decreases as we approach the equator and then increases on the other side of the equator. And this was found both with the field measurements in B and the museum measurements in C. We see this very clear pattern supporting Bergmann's rule. So what about with frequency, with song frequency? So we go over to A here. Now, there may be differences among the species in frequency, certainly taxon-based differences in frequency. So here we've focused on the two species with the widest distribution, the widest latitudinal range, and that's the yellow-fronted and red-fronted tinkerbird. And what we see here is that indeed the lowest frequencies are at those higher latitudes. And then we see this increase in frequency towards the equator and decrease on the other side. It's not the highest frequencies are not exactly at zero at the equator. However, we attribute that to some of the differences in elevation. A lot of our equatorial sites are at higher elevations in Uganda, Kenya. And so then this is based on the raw data and just looking at correlations with latitude. In the actual results of our models, the statistical tests show that we see those patterns both in morphology, here in the field collected measurements, and at the top there we can see that as latitude increases, body size increases quite clearly, very highly significantly, with a very large effect size as well, marginal R squared. There's also a significant effect of elevation, and also in beak length we see a weaker effect, p-values barely significant there, for latitude on beak length, on relative beak length, and also of elevation, slightly non-significant. But a clear effect of Bergmann's rule, not so evident Allen's rule on morphology. Looking at song frequency, and there was a similar pattern with the museum specimens as well. In fact, even less evidence of Allen's rule with the museum specimens. Looking at overall at song frequency, it significantly decreased with increasing latitude and also elevation. But there was no effect of vegetation density on peak frequency. However, patterns at the taxon level were especially revealing as well. I don't show them how it's what I showed you before with the yellow-fronted and red-fronted tinkerbird. So we have a very clear relationship between latitude and elevation, body size, and also song frequency. However, those data sets are independent data sets. It wasn't the same birds that we measured, that we recorded, certainly not from the museum specimens, we didn't record their songs. So if we look at the hypotheses for the things that may affect vocal frequency, we have this morphological constraint hypothesis that there's this negative correlation between body size and song frequency. There's also the widely recognized acoustic adaptation hypothesis that there's this negative relationship between the density of the habitat and song frequency. And there may also be genetic differences that may explain some differences we have in song frequency among different species. Now, given the study that we have here and the opportunity to look at across a hybrid zone, if you have consistent differences between species, you might expect that admixed individuals will produce intermediate song frequency. So this is something to look at as well. We've looked at, across this contact zone of red-fronted and yellow-fronted tinkerbird, we've looked to see directly, using the same individuals that we've ringed and then subsequently recorded, look to see if we can see what the direct effects are of morphology and other characters and environmental effects on song frequency. And so this is based on fieldwork performed over the last 8-9 years, where we've ringed, measured, sampled the DNA of, and recorded these colouring birds singing. In this case, we managed to get a sample size of 106 individuals and 382 recordings from them across this hybrid zone, where we have birds in the contact zone there, in this sort of bluish shade, and then in Alopatri going into South Africa on either side into Mpumalanga and Gauteng, and red-fronted into KwaZulu-Natal. And for each bird that we've captured and recorded, we've identified the vegetation and climatic data associated with that point. So identify what the vegetation structure is using these metrics from the MODIS satellite, that's the Enhanced Vegetation Index, the Leaf Area Index, and the percent tree cover. We've also obtained elevation from the Shuttle Radar Topography Mission, and from WorldClim, the Bio1, the mean annual temperature and annual rainfall, and we've also obtained humidity to look at their effects on song frequency. So you can see some maps there of those distributions of elevation, temperature, and rainfall. Yeah. Okay, and so there's certain hypotheses, and the way we wanted to look at this is to look at both direct and indirect effects of the environment on song frequency. And so there are certain predictions that we have. I mean, we talked about the Bergman's rule effect on how temperature may affect body size and how that may in turn affect song frequency. But temperature may also have direct effects on song frequency as well. There are some of these other climatic effects may affect body size. Ancestry certainly may affect body size, but may also affect song frequency directly. And then the vegetation structure is expected to have a direct effect on song frequency. Looking first at the patterns, we can see very clear pattern of frequency with temperature. So increasing temperature, increasing frequency. There's also a pattern with rainfall. And the expected strong pattern with body mass, of decreasing frequency with body mass. And there's also a pattern with humidity, of increasing frequency with humidity, with increasing humidity. And we can also see ancestry mapped onto here. So you can see there appears to be a pattern with ancestry here as well. And just looking at correlation, that is a highly significant pattern as well. We don't see any pattern with vegetation density in any of the metrics that we've looked at. There's no significant pattern there at all. Now, using path analysis, we reveal the direct effects. And so looking first at the possible indirect effect, we see that there's a strong correlation, the strong effect of Bio1, that is mean annual temperature, negative effect on body mass. So the lower the temperature, the higher the body mass. That was what we predicted, and it's very clear from this analysis. And then what are the effects on peak frequency? Humidity has a positive effect. There's a negative ancestry effect, and there's a negative body mass effect. And so body mass, which is affected by temperature, affects peak frequency. There's no direct effect of temperature or of any of the other variables that we looked at. And so looking at the path of significant effects, what we see here is that there is a direct effect of ancestry, of humidity, and of body mass, which in turn is affected by mean annual temperature. So we have support for the morphological constraints hypothesis, driven by Bergman's rule effects of temperature on morphology. There is no support for the acoustic adaptation hypothesis. Okay, so that's looking mostly at environmental effects, but we did see an effect of ancestry. So let's look more closely at genetic effects, this time focusing on some other aspects. First, I'll give you a little bit of background on identifying genes associated with certain characters. So many genes have been identified that underpin morphological characters. For instance, feather color and also bill color. One gene implicated in carotenoid conversion is CYP2J19. This is on chromosome 8 in birds, and it's been identified as the enzyme that catalyzes the conversion from yellow dietary carotenoids to red ketocarotenoids. And that's in feathers, it's shown in the lab in canaries and also in the bill color of zebrafinch. The genomic associations with behavior can be more challenging to identify, but there has been a study identifying a gene associated with migration distance in peregrines. And a lot of work has investigated the genes associated with vocal learning. And that's in those three orders that we talked about before, the Osing passerines, the parrots, and the hummingbirds, and the genes associated with vocal learning in those species. But what about the genomics of non-learned vocal function? Can we identify the genetic basis of how fast a bird sings? And which genes are involved? And are there known functions of these genes in human communication? Vocal learning can complicate the discovery of vocal control genes. So ideally, we would focus on a system with non-learned song that's simple in structure. Okay, so we're focusing on rhythm here. What do we know about rhythm? Well, our beating heart beats at a constant rhythm. Rhythm is something that transcends birdsong. Here's an example of a nightingale and its song. You can see a spectrogram of its song there with a lot of different rhythmic components to it. And of course, rhythm transcends human culture, dance, music. So we've mentioned these the groups of birds that are known to learn their songs. And if you look at the brain structure of these groups, things like songbirds and parrots, they have certain structures in the brain that are similar to those structures in humans, play a role in learning of sounds, of learning songs, learning language. And these are reduced or absent from organisms that don't have the ability to learn vocalizations. And so we can see that here they're absent from the vocal non-learner, the sub-Osing group here, and also from these other orders of birds, pigeons, families, quails that lack those structures. And that's the case for tinkerbirds as well. This is where tinkerbirds come. They're Pisciforms related to woodpeckers. And as we showed before, we have pretty good evidence that they learn their songs rather than develop innately. Okay, and so we're focusing here on the red-fronted tinkerbird and yellow-fronted tinkerbird again. No, do not learn their songs. This is the distributions of these two species, and they come together with these contact zones that are illustrated in blue. And at these contact zones, they hybridize. Hybridization provides us with the opportunity to identify the genes that underlie phenotypic characters. We focused this study in Southern Africa, across that Southern Africa contact zone that we talked about before, where we'd previously shown the extent of first attempts to look at the extent of hybridization, where we identified it. This was using microsatellites, and a study led by former PhD student Emmanuel Mwanko. And we can see here, so each bar represents the ancestry of an individual, and the more yellow it is, the more yellow-fronted ancestry, the more red-fronted ancestry is. As you can see in the sympatry zone there, that you have lots of individuals with mixed ancestries, lots of evidence of hybridization. And because of this extent of hybridization, this allows us to identify characters that might underpin the genes that might underpin certain characters. And we first found an association with that same gene, CYP2J19, that had been identified as being involved in the conversion of dietary carotenoids to red ketocarotenoids. So we identified that first time in a wild population across this contact zone in Southern Africa. And we can associate that with the different carotenoids as well. So in the top right here, we can see Raman spectra here, which identify the differences in the carotenoids between the yellow-fronted tinkerbirds and red-fronted tinkerbirds. So this study was based on the individuals that we caught in the hybrid zone. That was 87 individuals at the time. So we captured them and, as before, recorded their songs, measured the pulse rate, these color ring birds across this contact zone that we mentioned before. And we focused here on the inter-onset interval. And so here we have the spectrograms of red-fronted and yellow-fronted tinkerbird songs, and also of a hybrid individual shown in the middle there. And here we're showing on the right, we're showing the measurement that we focused on, this inter-onset interval, which is the time between the start of one note to the start of the next note. And we did this for the entirety of the recordings we got. And so for each recording we got, we got about 200-300 inter-onset intervals, calculate the mean for each individual for those values. We then sequenced the entire genome of the individuals that we caught, and we mapped them onto the Tinkerbird reference genome that we assembled with the Vertebrate Genome Project. And we identified which part of the genome is associated with how fast birds sing. So here again are spectrograms and waveforms of the two species songs. And from this, you can probably start to deduce what the main difference is in the speed. And you'll be able to hopefully hear it on these videos that I'll play now. This is a yellow-fronted tinkerbird. And a red-fronted. I don't know if you could hear that difference, but there is a subtle difference in how fast they sing. The red-fronted sings a little bit faster. And there's no overlap between the speed of those songs and allopatry. So as I said, we focused on this inter-onset interval across these recordings. And we first identified that these songs are isochronous. What that means is that the notes are given with an even spacing. So essentially they're living metronomes. And so this character isochrony is actually quite rare in vertebrates. It's only been identified in primates, in bats, and in two songbirds prior to what our identification here in Tinkerbirds. And so then we examined the association between inter-onset interval, the pulse rate, with the genome. And so this is a Manhattan plot that's showing which region of the genome is most closely associated with the trait. And so we can see this area here above this dashed line. This is the area that's significantly associated with how fast these birds are singing. And there are a number of genes that these SNPs map onto. In particular, neurexin-1 has had 3 SNPs that mapped onto it, and these other ones here that we've identified as well. What do we know about these genes? Well, neurexin-1 is expressed in the brain, and it has previously been associated with speech disorders in humans, also with migratory behavior in passerines. COC8A is expressed in skeletal muscle, and it's also been associated with vocal impairment in humans. ENAH is expressed in the nervous system and has been associated with hearing disabilities in humans. MSH2 has not yet been associated with a function. We then looked at genome-wide ancestry, identified the genome-wide ancestry of each individual. And so on the left here, we can see the genome of each individual, so that's each chromosome, and the ancestry across each chromosome. And this has then allowed us to look at the regions of interest in each individual's ancestry at and genotype at those loci of interest. And we can also see that looking at genome-wide ancestry, we can see yellow-fronted tinkerbirds are singing these slower songs with these higher inter-onset intervals, red-fronted the faster songs. And as we go, we look at admixed individuals, we can see that they're singing mostly intermediate song in terms of pitch, of pulse rate compared to the parental species. So what effect does hybridization have on the rhythm of these birds? Does it affect its metronomic quality? So, I mean, we've shown that on average that those hybrids sing intermediate song, but does that mean it's an intermediately paced throughout, or is it a mixture of, sometimes it sings a bit faster, sometimes a bit slower, or is it sort of more unstable? Well, there are certain hypotheses of how pitch may be affected by hybridization. It could be that it could affect the mean of the trait, as we saw in that last plot, that we have the mixed ancestry individuals would have an intermediately paced song. It could also affect the variance and the stability of the songs. Maybe they're singing this sort of more, mixture of faster and slower songs. It's more unstable song. So we investigated that as well. And we also investigated whether hybridization could lead to character displacement in some parental species. If we have a hybrid zone, hybridization, is there any reinforcement against hybridization? Could this be manifested in character displacement, either the mean or the variance of the song, with the predictions that as you approach the contact zone, so as you go towards the left here, that the songs would be more different in the contact zone between pure red-fronted and pure yellow-fronted tinkerbirds, and in variance, that they'd be less variable as you approach the contact zone, the hybrid zone. So we use these double hierarchical generalized linear models to investigate these hypotheses. And what we found was that not only is the red-fronted tinkerbird song, so the proportion Pazillus here is indicating how red-fronted tinkerbird it is, not only are the songs a lot faster, they're also more stable. So the blue box here shows there's significantly more stable song, less variable song, lower variance in red-fronted tinkerbird song compared to yellow-fronted song. But above that, we don't see a significant quadratic effect, which is how we were looking at this variance effect of hybrids. We don't see that there. However, if rhythm is affected more by specific genes as opposed to genome-wide ancestry, you might expect that it's at those loci that have an effect on rhythm, that we might see an effect of hybridization. And we do indeed see a significant effect here, quadratic effect, on neurexin-1 and coc8a. Those are the two genes that were shown to, previously shown to impair human speech. We see in those that mixed ancestry individuals have less stable songs than red-fronted tinkerbird. And we also find that there are greater differences in stability between pure Poecilus and pure Exili in the contact zone compared to allopatries. There's also character displacement in stability, not in the actual trait itself, but in the stability of the trait in the contact zone. So there's character displacement. Hybrids at the particular loci of interest sing less stable song. Does this affect, is this an indication that there might be a sort of mating in that zone? Well, we looked at this by examining the ancestries of individuals. So to look at sort of mating, you want to be able to see what the ancestry of the male is and the female of a pair, but we didn't have these data. We haven't been tracking these pairs for years to see who's mating with whom. What we have is individuals caught and the genomes of those individuals. So we wanted to be able to infer what the mother and what the father is of individuals that we caught. And we went about that by looking at the females. We could investigate that in females because females are the heterogametic sex. So they have ZW sex chromosomes. And we assumed that the father's ancestry would be equal to the Z chromosome ancestry of those females because they inherit the Z chromosome from their father. And then from there, we could then calculate the likely maternal ancestry, the mother's ancestry. And then in this plot in the middle here, we map those values that we calculated, the mother's ancestry and the father's ancestry. And what we can see here is if we look at pure red-fronted tinkerbirds and pure yellow-fronted tinkerbirds, who they're mating. And so we looked at those mothers. Who are they mating with? The red-fronted mothers there. This is looking at the top 3% in terms of ancestry, 97% red-fronted and above. And what we can see here is that the pure red-fronted tinkerbird females are mating with males that have a very high proportion of red-fronted ancestry. The yellow-fronted females are mating with males of any ancestry. So it seems as though the red-fronted females are very choosy. They only want to mate with red-fronted males. The yellow-fronted, not so much. They're happy to mate with any male, even hybrids. So the pattern suggests there is asymmetric assortative mating. Female red-fronted tinkerbirds mating only with males with a high proportion of red-fronted ancestry, which is those males that sing faster, more stable songs. However, those males also have red foregrounds. Now, from the work that we've done with feather color, we know that the red allele is a dominant trait. And so all these hybrids here, they have red foregrounds as well. But the females, the red-fronted females, are not mating with these hybrids. They're only mating with those individuals with a much higher proportion of red-fronted ancestry. So what this suggests is that those females prefer males that sing fast songs, or they prefer males that have fast songs and red foregrounds, combination of the two. This is something that we're investigating further, but certainly yellow-fronted female times red-fronted male is much more common than the other way around. And so there's a picture from the hybrid zone, mostly in Eswatini. Okay, so when a female hears a song of a male like this individual here, is she attracted to the foreground color or the speed or stability of the song? These are questions that we're continuing to investigate. And we're doing this by performing experiments in the field, looking at responses to differences in song and foreground color using robotic models. So we started doing these experiments on our last field trip in November and December. With, in this case, we had some undergrads in the field with us. And this is what an experimental setup looks like. This is a 3D printed model that we've stuck on a robotic device that we're wirelessly controlling and making it move. And so we give them a choice of a red and yellow fronted Thinkerbird and play different songs. And we had another setup where we also have the throat moving. And so this is something that's very much a work in progress that we're looking to improve our experimental setup. Okay, and with that, I would like to thank, again, the members of the lab that have helped along the way with this work, our collaborators, in particular for a number of years now, Alan Brailsford, Bridget von Holt, Niels Dingemans, who helped us with the song stability stuff, Eric Jarvis for the genome assembly, our local collaborators in Eswatini and South Africa, our Monadium and Colin Downs, our funding sources, and also the landowners, the field assistants, the permit authorities that facilitated all this research. You can read our paper on vocal rhythm in Nature Communications there. And I'd like to thank you all for attending and again, Ellie, for the invitation. With that, I'd like to ask if you have any questions.
Donc, bonjour tout le monde. So, thank you, Professor Kirschel, for this wonderful presentation. So, I will just summarize in a few sentences the main points, and so I will do it in French. Donc, aujourd'hui, donc notre invité, il a présenté son travail sur plusieurs années qui concernent l'étude du chant chez les barbillons. Donc, c'est une espèce d'oiseau qui vit en Afrique. C'est une espèce qui est non nocine, c'est-à-dire qu'elle n'apprend pas son chant. C'est un chant inné. Et donc, il s'est intéressé à comment, entre les différentes espèces, donc l'environnement et la génétique, va influencer sur les caractéristiques, notamment fréquentielles, des chants de cet oiseau. Et donc, une des premières expériences qui avait été menée, c'est en étudiant, il y avait 3 espèces de barbillons le long de la côte est et la côte ouest en Afrique, avec comme hypothèse donc, que le bruit de l'océan le long de la côte, qui est donc un bruit d'ambiance, allait contraindre, entre guillemets, les oiseaux à modifier leur plage fréquentielle afin d'être mieux entendus. Et donc, il s'était aperçu que plus on s'approchait de la côte, plus la fréquence dominante, la fréquence fondamentale, qui est aussi la fréquence fondamentale du chant chez ces oiseaux, allait devenir plus haute pour justement passer au-dessus du bruit de l'océan. Et donc ça, c'est intéressant parce que ces adaptations fréquentielles, on les avait observées chez des passereaux, par exemple, qui vivent en ville. Donc c'est cette capacité de modifier le chant. Mais dans le cas des oiseaux qui apprennent leur chant, ils ont cette capacité de pouvoir, en apprenant à modifier leur chant. Mais par contre, chez les oiseaux non nocines qui apprennent pas leur chant ou c'est inné, c'était beaucoup plus rare d'avoir ce genre d'exemple où il y a aussi un impact environnemental sur les modifications du champ. Donc ensuite, ils avaient aussi trouvé que dans les zones de sympatrie où donc des espèces de barbillons allaient cohabiter, ils avaient tendance à avoir des champs beaucoup plus distincts en termes de fréquence dominante, c'est-à-dire que leur fréquence dominante allait s'éloigner pour empêcher justement qu'il y ait un masquage entre les champs des différentes espèces. Il s'est aussi intéressé à d'autres variables environnementales, comme notamment la température, la latitude en Afrique où ces oiseaux vivent, Et en gros, il y avait deux modèles théoriques. Il y avait le modèle enfin, la règle de Bergman qui démontre qu'il y a un lien entre la masse corporelle de l'oiseau, la taille et la masse de l'oiseau et sa fréquence. C'est-à-dire que plus l'oiseau est gros, plus sa fréquence de chant va devenir basse, et plus il est petit, plus sa fréquence va devenir haute. Et l'autre modèle, l'autre règle, c'est la règle de Allen, où là, c'était plutôt par rapport au bec. Plus le bec est petit, plus la fréquence augmente. Et donc, il a créé des modèles où il a inclus plusieurs variables environnementales pour essayer de voir justement quelles sont les variables qui vont impacter sur la fréquence dominante chez les oiseaux. Et donc, ce qui ressort principalement, c'est que plus la température augmente, plus les fréquences aussi augmentent. Et de manière générale, c'est avant tout la taille des individus qui va être modifiée et qui va aussi impacter leur fréquence. Donc, plus la taille de la masse de l'individu augmente, plus sa fréquence diminue. Il y a aussi un lien avec l'humidité, que la fréquence du champ augmente avec l'humidité, mais par contre, il n'y avait pas de lien avec la végétation. Et à part les effets des différentes variables environnementales, il y avait aussi des expériences qui ont été menées, une étude sur la génétique, comment la génétique impacte aussi les paramètres acoustiques et aussi la couleur du plumage de ces oiseaux. Et donc, pour cette partie, ils se sont intéressés notamment au rythme, puisque donc, ces oiseaux ont la particularité d'avoir un rythme vraiment très stable. Donc, ils chantent à une vitesse très bien définie entre les différentes espèces et le rythme est très stable. Et du coup, ils s'étaient intéressés notamment à comment ce rythme et la vitesse de chant et les distances entre les différentes syllabes évoluent dans les zones d'hybridation où on a des hybrides. Comment, en fait, le fait qu'il y ait une hybridation, cela va impacter les caractéristiques du chant et si les femelles vont être sensibles à certaines caractéristiques ou pas, et comment elles vont se comporter face à un mâle d'une autre espèce, ou en tout cas un mâle hybride issu de deux espèces proches. Et pour finir, donc en ce moment, ils sont en train de tester euh, des robots qu'ils ont fabriqués, donc où ils vont reproduire des variations de couleurs au niveau du front pour justement en apprendre un peu plus sur ce phénomène d'hybridation et la relation entre les caractéristiques du champ, l'hybridation et la façon dont se comportent les mâles et les femelles, donc avec des petits robots qu'ils ont fabriqués, qui ont différentes couleurs et qui vont produire différents types de sons, et donc ils vont voir un petit peu la réaction des oiseaux face à ces robots. Voilà, et maintenant ça va être le moment des questions. So thank you for your attention, and now you are free to ask any question you want.
There's nothing left, all gone and run away. Maybe you'll tarry